Good morning, AP Biology students. Today we're talking about fluid, acid, and bases. So more specifically, our subject today is chemical buffer systems against changes in hydrogen ions, so H+. So what are the different chemical buffers that are out there? So there are 4 different types of chemical buffers, and each chemical buffer has a primary role. I will identify the major functions of each of these 4 buffer systems. Then in the next few slides, we'll go in depth on how they carry out these main functions. So the first chemical buffer that we'll talk about is carbonic acid and bicarbonate. So that buffer pair. This buffer system's main role is to create a buffer in the extracellular fluid, which is the fluid surrounding the cells, such as plasma. It works as a buffer against the non-carbonic acid changes. So the next buffer we'll talk about is the protein buffer system. And the role of protein buffer is to mainly buffer intracellular fluid, which is the fluid inside the cell, but it also additionally buffers the extracellular fluid. The third buffer system we'll discuss is the hemoglobin buffer system, and this is one that we've talked about previously when we discussed blood in the hemoglobin. So the hemoglobin buffer system against carbonic acid changes. So the last buffer system is the phosphate buffer system. This buffer system also buffers the intracellular fluid, just like the protein buffer system. But the big main difference with the last buffer system is that it plays an important role in the urinary system because it is a urinary buffer. So I have these different buffer systems laid out here: intracellular fluid, extracellular fluid. All right, so I can throw these big buffer systems at you, but what do these buffer systems do? These buffer systems work to minimize any changes in the pH by binding with hydrogen ions or yielding free-floating hydrogen ions. So chemical buffers are composed of 2 chemical compounds, so they create a mixture in the solution. So you'll notice while we are talking about the buffer systems that each of these buffer systems are part of some type of chemical reaction and that they can be reversed. In the chemical reaction, there will be a chemical substance that will yield the free hydrogen ions, and then the hydrogen ions will decrease, causing a more alkaline environment. And there's a substance that will bind to the hydrogen ions, and then there'll be a high amount when there's a high amount of them. When there's a higher amount of hydrogen ions, that means that the solution is more acidic. And when there is less hydrogen ions, that means the solution is more alkaline. So I'll show you a pH scale further on in our slides to kind of show this acid and base scale. So an example to demonstrate what this looks like can be easily shown in our first type of buffer system, so that we'll discuss, and that is our H2CO3 or HCO2-3. So this is our bicarbonate carbonic acid buffer system pair. So the picture to the right is the reversible reaction for carbonic acid bicarbonate. So that's over on this side to the very right side here. The CO2 and H2 that comes into play later with one of our other buffer systems. But I really do love having that whole reaction because this is something that we've already seen in a previous lesson. So kind of reiterating that. If there were some type of strong acid that's going to be added, so a hydrochloric acid to a solution of this, if we're adding it to a solution of the with the hydrogen ions, would end up binding to this HCO3 to form H2CO3. This will only happen if the solution contains carbonic acid, so that carbonic acid and bicarbonate pair. Without this buffer pair in the solution, the strong acid would cause hydrogen ions to remain free in the solution, When looking on the flip side of the solution, if it has a loss of these hydrogen ions, the solution will become more alkaline, and the buffering pair will release hydrogen ions from H2CO3. As previously noted, this buffer pair's main role is to buffer extracellular fluid. The extracellular fluid has both a lot of H2CO3 and HCO3. So to help keep the pH at an equilibrium. This buffer pair is overlooked by both the kidneys regulating HCO3 and the respiratory system regulating the CO2. In the full reaction, so on the left side, you can see that the CO2 created the H2CO3, which we discussed, again, we discussed this in our respiratory unit. So an example on how this works, think about when you're exercising, for example, when I like going for runs, my muscles will sometimes get really tired and I can feel them get sore, which is that lactic acid. And when that lactic acid, it goes into that extracellular fluid. This means that there's an increase of hydrogen ions there. So this buffer system is going to kick in, and the buffer HCO3 will bind with these hydrogen ions that are created from this lactic acid, so that we don't continue to get a buildup of those hydrogen ions. So this is an example other than the CO2 initiating the change. Something, again, that we've seen before. An example for the reverse side is if we lose hydrogen ions. So I like to think about when you go on a roller coaster ride at Six Flags and your stomach's not feeling so great and you vomit. And this will cause that stomach acid to come up when you get sick. And what happens is that those H plus ions, if they're accumulated in your stomach, you're getting rid of them. That's acid, and we need that acid in our stomach. We need some of that acid in our stomach. So what's going to happen is CO2 and H2O will make more H2CO3, which will create more of those hydrogen ions to come back that we lost when we got sick after that roller coaster ride. So buffer systems cannot buffer themselves. If there is a respiratory problem causing an increased amount of CO2, this will lead to those hydrogen ions to accumulate in the plasma. So that's acidosis. This will push the reaction to the right side because of the CO2 increasing. Same thing on the opposite side. A reduction in CO2 causing a loss of hydrogen ions, so alkalosis. So you'll see the rest of these buffer systems that we talk about have some of those similarities that we see with this same reaction or process here. So when you're looking at the protein buffer system, it's important. This is an important buffer system to the intracellular fluid. This is because there's many proteins in the intracellular fluid, especially in comparison to the last buffer pair we discussed. Although there are less plasma proteins than intercellular proteins, this is still one of the larger buffer systems in our body fluid. So what you can see here on what's being, who's taking in the hydrogen ions and what's giving up the hydrogen ions. So in the case of the protein buffer, it's the carboxyl group that is giving up the hydrogen ions, and the amino group that's accepting the hydrogen ions. And that's what this image is showing you guys in the chart here. All right, so hemoglobin buffer system. So when H plus, so the hemoglobin buffer system buffers H plus when CO2 is brought into the tissue and the lungs. The CO2 will move into the blood, causing an accumulation of CO2 and H2O. So this will create HCO3 and H plus, that hydrogen ions, when the enzyme carbonic anhydrase initiates this reaction. So it's going to speed that up. but hemoglobin will reduce most of the hydrogen ions by binding them to it. It will then carry it away so that there's no longer affects the body fluid. If it wasn't for hemoglobin, the blood would become extremely acidic during its process of transporting that CO2 to the tissue. This buffering capacity that the hemoglobin system has maintains that acidic level in the venous blood. However, the venous blood is more acidic than the arterial blood. And this reaction, again, if we're reversing it, we can see this when the CO2 is exhaled and when O2 is breathed in. So again, it's that reverse process. So we can see this process. I really like this image. Again, this is an image that we've used in a past lesson. So you can see when their O2 is brought in, it's going to have that hemoglobin is going to bind to hydrogen ions. And then when we breathe out carbon dioxide, again, you can see this H plus come off here too. So again, a really great chart to reiterate that. So the last buffer system that we will discuss is the phosphate buffer system. This buffer system is made up of acidic phosphate salt, so NaH2PO4. This acidic phosphate salt can provide additional hydrogen ions when hydrogen ions are low, and it can also accept hydrogen ions when hydrogen ions is too high in the body fluid. So just like we've seen in all of these chemical buffer systems already. So the buffer pair is alternating the hydrogen ions and sodium ions depending on the amount of hydrogen ions present in the body fluid. This buffer pair is more important in the intracellular fluid because there's more phosphate inside of the cell. So this buffer pair is also a great urinary buffer because as humans, we like to consume more higher amounts of things than we need. So we consume way too much phosphate. So when we do that, it's got to go somewhere. So this extra phosphate is stored in the tubular fluid until it's excreted from the body. And this is done through the filtration from the kidneys. So this is the only chemical buffer system that is present in this tubular fluid, and it helps buffer the urine as it forms. For other chemical buffer systems, you can see this HCO3 and CO2 are reabsorbed, whereas in the hemoglobin and plasma proteins, they're not filtered. So some compare and contrast between these different buffer systems. Something neat to look at is the Henderson-Hasselbalch equation can be used to determine the relationship of hydrogen ions and the buffer pair has. So to sum this up, it looks at the pH of the solution to the acid dissociation constant of the acid. And we'll take a day to look at this equation and do a couple practice ones as well, but that's the basis line for this equation. So the four chemical buffer systems that we have just discussed are the first line of defense against changes in hydrogen ions. These systems work right away. The second right away against, although phosphate buffer system is slower in comparison to the others, it still works very quickly. Therefore, when any of these buffers see that there is an altered change in hydrogen ions, the reverse chemical reaction will move either left to right, depending on if there is an increase or decrease in hydrogen ions present. So these chemical buffer systems are known for the first line of defense because they only respond first. It's kind of just like a first responder. If there's an accident that happens, that first responder comes in before the ambulance. That's what I like to think about. These chemical buffer systems are really like a first responder. They're first to the scene. So the chemical buffers take the hydrogen ions out of the bodily fluid. They don't completely eliminate the hydrogen ions, but from the body, the buffer pairs of these systems have hydrogen ions incorporated into them to either take in the hydrogen ions or they produce it out of them. So we need other defense mechanisms because otherwise our buffer chemical systems will not be able to buffer hydrogen ions because they would be full. They'd be bound already with those hydrogen ions. This is why the respiratory and renal mechanisms are incredibly important because they help eliminate acid from the body. Again, these systems are the second defense because they respond much slower. So again, respiratory and renal mechanisms, they're the secondary defense. So thank you guys for tuning in today, and we'll discuss some of these secondary defenses against changes in hydrogen ions in our next lesson. But before we depart today, I do want to share an interesting study done by Daniel P. Heal. And this study looked at acid and bases balances and with the hydration status, allowing consumption of mineral-based alkaline bottled water. So what they did, this study is so interesting. It looked at how consuming bottled water so an alkaline based bottled water can affect our buffers, how it can affect our pH. But additionally, with learning about these buffer systems, I think about how did these affect the buffer systems? Because obviously when we're changing the pH with our body fluids, these buffer systems are going to react to this. So it's kind of interesting to look at. So in the study, he looked at 38 individuals with varying genders and physical activity, and then split them into a control group and a experimental group. So the control group did not receive alkaline bottled water. Instead, they had a placebo bottled water. And they concluded results by using blood from fingertips and urine samples so blood samples from the fingertip, and they collected urine samples throughout the weeks of the experiment. It was about 4 weeks. And they looked at osmolarity, pH, and the total urine volume. The results showed that there was no change in the dependent variables for the control group. The experimental group, on the other hand, showed increases in blood and urine pH. So the blood pH went from 6.23 to 27.07. Oh, I'm sorry, 7.07. So 6.23 to 7.07. And the urine pH went from 7.52 to 7.69. For the osmolarity in the experimental group, the blood osmolarity decreased, but the urine osmolarity increased. Additionally, there was a decrease urine output, and it went from 2.51 to 2.05 liters per day. So in their experiment, they had their experimental group switch over to the placebo water in the 4th week to identify if the change would revert back to those set points or that starting point, which the data did show that there was a reverse in changes. So that was really interesting. And then in the conclusion of the study, they concluded that drinking alkaline water had a value. It was a value of influencing that pH balance and hydration in adults. So very, very interesting study. Thank you for tuning in. And if you have any questions, please stick around.